Okay, great. So thank you very much. I would like to thank the organizers as well for this invitation. I'm very sorry that I cannot be with you this year. Fortunately, other commitments have kept me from coming to Panama. But I'm very happy to even join you virtually and present our work, which has to do with the molecular mechanisms linking aging and neurodegeneration. Now, why is that interesting for us? It is interesting, especially in the context of neuronal cells, because as you've heard, neurons are complex cells. They depend on mitochondria for their energy demands. They are highly differentiated cells, so that means that they need to traffic their mitochondria across long appendages, axons, dendrites, etc. So this is a complex process, of course, and it depends on energy. These neurons are consuming energy even if they are sitting there idling, doing nothing, just because they need to keep their plasma membrane polarized, and this, of course, requires a lot of energy. But one other interesting aspect about neuronal cells, which is actually the main characteristic of them that has prompted us to look into energy homeostasis, specifically in neurons, is that neurons are the most long-lived cells in our bodies. So most of them actually are born even before we are born, and some of them will die literally after we are dead. So they will have to survive for tens of years, even more than 100 years if we are lucky. And we were really interested in understanding what kind of elaborate homeostatic mechanisms these cells possess in order to be able to cope with their energy demands for such a long period of time. Maybe we could learn something from these neurons, how they do it, how they manage. And of course, as was said, neurons depend on mitochondria for their energy demands. And actually, if mitochondrial homeostasis is not maintained in neurons, then of course we have devastating consequences such as neurodegeneration. And one of the hallmarks of Alzheimer's disease neurons in patients suffering from Alzheimer's disease is the accumulation of mitochondria, of damaged, mostly abnormal, misshapen mitochondria. So this is one of the features of Alzheimer's disease neurons. But it's not actually just observed in Alzheimer's disease. This feature, this accumulation of damaged mitochondria, appears to be a common denominator of many pathologies in humans. Some of them are actually very strongly, very tightly associated with aging. For example, in addition to Alzheimer's disease, of course, Parkinson's disease, Huntington's disease, other metabolic diseases associated with aging, diabetes type 2, obesity, cancer, ALS, etc. Those are all diseases that have been associated with aging. And as a common denominator, we observe accumulation of damaged mitochondria in the context of these pathologies. Now, even more interestingly, in the context of normal aging, but not just in the context of disease now, but in the context of normal aging, we observe progressive accumulation of damaged mitochondria during aging. So, for example, in rats, this has been observed using electron microscopy. We can even use this type of observation to count mitochondria in cells. We can compare 6 months versus 24 months of age. There is a clear increase in the number of mitochondria in cells. And this phenomenon is evolutionarily conserved. So we don't just observe it in mammals, we also observe it in nematodes, in Sinorhabditis elegans, which is a very popular model organism to study aging and neurodegeneration. And here, actually, we can even monitor this accumulation in vivo using specific fluorescent reporters that contain mitochondrial localization signals. So GFP is localized to mitochondria, and then we can use fluorescence as a proxy for the number of mitochondria. And the more mitochondria we have, the greener a cell becomes a tissue, and we can even use tissue-specific promoters to drive the expression of these reporters in any type of cell that is of interest to us, for example, neurons. In this case, I'm showing you intestinal cells, the C. elegans intestine. And if you compare day 1 of adulthood versus day 10, which is quite old for these animals already because they live for about 15 days, again, we observe the same accumulation that we've observed in mammals. So this has prompted us to study the underlying mechanisms, because if this phenomenon is evolutionarily conserved, then most likely the mechanisms that bring about this phenomenon are also evolutionarily conserved. And to do this, we took advantage of the features of C. elegans, the experimental malleability of the nematode, which is the best characterized animal that we can use in research even these days. So we know exactly, for example, how many cells there are in the adult, 959 cells. We can even follow all cellular divisions from the zygote down to the adult. And this is, you can see here, the animal is transparent. We can record all cellular divisions. The embryonic development takes about 14 hours, and then the adult becomes fully developed after 4 larva stages after hatching. But as you can see, we can visualize all cellular divisions. This, of course, gives us important advantages, because we can target specific cells. We can target specific neurons. And when it comes to the nervous system of the animal, again, we have unparalleled detail of characterization. C. elegans is the only animal for which we have the whole circuit of the nervous system. So we know exactly how many neurons there are, 302 neurons. We know how they are connected to each other, 6,000 synapses total. And we can even follow signals through the nervous system. So from the sensory neurons to interneurons to motor neurons. And this gives us, of course, the opportunity to do experiments that are simply impossible in other model systems. Now, because of these advantages, C. elegans has been pivotal in understanding aging. And actually, the first genes that were found to regulate aging were isolated in C. elegans, and then their homologs and orthologs were identified even in humans. So, for example, through these studies, we now know that the insulin signaling is involved in regulating aging. The first genes were isolated in C. elegans and characterized there. Dietary restriction, mitochondrial function, the reproductive system, metabolism, the TOR pathway, genes controlling biological rhythms and either ultradian or circadian rhythms, stress response pathways, genetic and epigenetic pathways are also converging to influence aging. And we don't just know what pathways influence aging, we can even use this knowledge to manipulate aging, to change the lifespan of an organism. And here, for example, you see survival curves. Wild-type animals, this is the black dotted line, is the survival curve for wild-type animals. You don't need to pay attention to all the lines. Just compare this black dotted line with this line with the red triangles. These animals actually have been modified in 2 ways. First of all, we dietary restrict them, so we don't feed them ad libitum. We restrict them the amount of calories they receive. And then we also, through genetics, through specific genetic manipulations, also down-regulate insulin signaling. And this is enough to extend the lifespan of these animals more than 6 to 8-fold. So instead of maximum lifespan 20 days, now we have a maximum lifespan of 140 days. Mean lifespan, instead of 15 days, now even more than 90 days, as you can see. This would mean that humans would be able to live, if we were able to translate directly these results to humans, humans would live 600 years mean lifespan. Maximum lifespan would exceed 1,000 years. Of course, things in humans are far more complicated, but actually most of these pathways are evolutionarily conserved, and similar manipulations will result
in lifespan extension in mice and even in primates. Experiments have been done with monkeys, chimpanzees, gorillas, and again, in those animals, we can extend lifespan, prolong health span using similar interventions. So most likely, actually, even through epidemiological studies in humans, these pathways, the same pathways have been implicated. Now, going back to the original question, which has to do with the accumulation of mitochondria during aging, what made it even more interesting for us to study this process was that in long-lived mutants, and I've showed you some examples in the previous slide, in C. elegans we can manipulate lifespan, so we can introduce specific mutations, for example, in a DAF2 gene, which encodes the insulin receptor in the worm, and DAF7, which encodes the TGF-beta molecule in the worm. Those two mutants are long-lived. And interestingly enough, in those long-lived mutants, we don't see this accumulation of mitochondria that we see in wild-type animals. So this suggested that maybe accumulation of mitochondria has something to do with the aging process itself. Maybe there is a causative relationship there. And in order, of course, to do this, we had to manipulate the content of the mitochondrial content of cells at will. And we can do this genetically in C. elegans. For example, we can introduce specific mutations in mitochondrial proteins called prohibitins, which will result in altering the number of mitochondria in cells. In wild-type animals, we exacerbate accumulation of mitochondria, so we have even more mitochondria in wild-type cells. Whereas in long-lived mutants, in DAF2 and DAF7 long-lived mutants, doing this exact same intervention even further lowers the accumulation of mitochondria in these cells. And then we can ask what happens with the lifespan of these animals. And what we've seen is that whenever we enhance the accumulation of mitochondria, we actually shorten lifespan, but whenever we suppress accumulation of mitochondria, so we keep the number of mitochondria low, we extend lifespan in these animals. So this became interesting, but of course we wanted to know what is the mechanism that brings about this change. What are the mechanisms essentially that control mitochondrial homeostasis in cells, that control the number, the content of mitochondria in cells? And if we consider what processes are involved, of course, on one hand, we have processes that result in the generation of new mitochondria. So we have mitochondrial biogenesis. And on the other hand, we have mitochondrial degradation. And in young, healthy cells, what we see is that these two processes are balanced. So the number of mitochondria in cells remains steady. Of course, there can be some regulation depending on energy demands. So mitochondrial biogenesis may temporarily become higher, or mitochondrial degradation may temporarily become higher, but eventually balance is restored, and that's how we have a controlled number of mitochondria in cells. However, during aging, and also during neurodegeneration and other pathologies associated with aging, this gentle balance collapses, and instead we now have a runaway accumulation of mitochondria. Now, I won't really go into all the details. We studied this process, and what we identified is that specific proteins involved in a process called mitophagy are actually playing a role in this balance, which then collapses and we have runaway accumulation of mitochondria. These proteins are called DCT1, NICs, PINK, and parking. And all three proteins are involved in a process, have been implicated in a process called mitophagy. Now, what is mitophagy? As the word implies, mitophagy is a specialized type of autophagy, actually a cargo-specific autophagy, that targets mitochondria. So mitochondria are degraded through this process. Mitophagy can happen in the physiological context, is actually a homeostatic process. So for example, whenever cells starve, animals starve, mitophagy is induced because you don't need mitochondria if you've got nothing to burn. And all this excess mitochondria is degraded through mitophagy. Also, during maturation of our red blood cells, mitophagy eliminates mitochondria. Our red blood cells, in addition to not having a nucleus, to being enucleated, also don't have mitochondria. And you don't need mitochondria in a cell that carries oxygen, has to transport oxygen to tissues. You would not want an organelle that consumes oxygen to actually be there. So that is why, very wisely, evolution has made it so that mitochondria are eliminated during red blood cell maturation. Now, all these are physiological processes, but also in the context of pathology. For example, whenever damaged mitochondria appear in cells, start to accumulate, then we have a specialized type of mitophagy that only targets damaged mitochondria, and this is controlled by the so-called pink parking pathway. Now, pink is a serine threonine kinase, parking is a E3 ubiquitin ligase, and through the activation of this pathway, mitophagy is locally activated in the vicinity of damaged mitochondria and eventually eliminates those mitochondria. Now, we characterize this process, the pink parking pathway, in C. elegans. We even identified the specific residues, amino acid residues that become ubiquitinated upon induction of mitophagy in the worm. And we've also looked at what happens if you downregulate very specifically a mitophagy in C. elegans. What we've seen is that mitophagy plays a very important role in lifespan extension under conditions of low insulin signaling. For example, DAF2 mutants live longer than wild-type animals, but this, to a large part, requires mitophagy, because if we downregulate mitophagy, we shorten the lifespan of these long-lived animals. And the same thing, of course, if we downregulate DCT1, a mediator of mitophagy, pink, or parking. But interestingly enough, in wild-type animals, if we downregulate mitophagy, we don't see any effect. Lifespan remains unchanged, as you see here. Now, of course, this, one might say, is expected, because in wild-type animals, under normal conditions, we don't have induction of mitophagy. However, this is paradoxical, and I will give you in a minute the reason why this is. In addition, we've developed a specific way to monitor mitophagy, to look at the mitophagy levels in vivo in live animals and cells, because this method is adaptable to other systems, for example, mice, cell cultures, other organisms, Drosophila, etc. So we developed a reporter that essentially is a fusion of GFP and DSRED, containing a mitochondrial localization signal. If this reporter is sent to be expressed in specific cells, it becomes localized in mitochondria, and if these mitochondria become substrates of mitophagy, then they end up in lysosomes where pH is acidic. And then we have quenching of the GFP fluorescence, the green fluorescence, whereas DSRED is immune to low pH found in lysosomes. So a dip in the ratio between GFP and DSRED signifies induction of mitophagy, specifically of mitophagy. And we can then monitor mitophagy away from other types of general autophagy. And what we found is that in long-lived mutants, such as DAF2, for example, or under stress, indeed, mitophagy becomes activated, as you see here. If you compare wild type to DAF2 mutants, or animals treated with CCCP, heat stress, or paraquat, an inducer of oxidative stress, again, all these stimuli can strongly induce mitophagy, and if specific mitophagy mediators are not there, such as, for example, DCT1, pink, and parking, then mitophagy cannot be induced, and we don't have this tip. Now, going back to neurons, for this last part of my presentation, what we've seen is that if you inhibit mitophagy, if mitophagy becomes downregulated, then we have a strong accumulation of damaged mitochondria in neurons throughout the nervous system of C. elegans. Now, of course, this creates strong oxidative stress upon these neurons, because now the accumulation of damaged mitochondria results in further increase of ROS production, and also in less ATP production, because the damaged mitochondria produce less ATP compared to healthy mitochondria. And as a result, we have lots of damaging consequences. In addition to increased ROS production, we have leakage of calcium out of mitochondria, we have collapse of the inner mitochondrial membrane potential, and less ATP being produced. So we have all these adverse effects if we downregulate mitophagy. Now, the paradox I was telling you earlier is here. If we indeed have all these adverse effects upon downregulation of mitophagy, why is then the lifespan of animals not altered? And this is a paradox because we know that lifespan is one of the most sensitive gauges of whatever is happening in a system, in a biological system, be it a cell or an animal or even humans. Lifespan, because it's a cumulative phenotype, it progresses through time, is a very sensitive gauge and is altered by almost any manipulation that we can impose. But here we don't see such an effect. So something must be compensating for these adverse effects that are caused upon downregulation of mitophagy. What is that? Now, one candidate process is the so-called retrograde signaling response. So whenever cells sense something is wrong with their mitochondrial pool, they initiate this retrograde signaling response, which is a signaling pathway from mitochondria to the nucleus, where most mitochondrial protein encoding genes reside, to induce the expression of these mitochondrial protein encoding genes and make fresh functioning mitochondria. This process is coordinated by PGC1 alpha, a master transcription regulator, in close coordination with other co-factors, transcription cofactors of the NRF family. Now, in C. elegans, we don't have a PGC1 alpha, but we do have NRF family members, NRF2 family members. One of them is SKIN1. This transcription factor is indeed required for mitochondrial biogenesis in C. elegans. Actually, if we look at the genes regulated by SKIN1, most of them are mitochondrial protein encoding genes. But interestingly, among them, there is also a master regulator of mitophagy. So it is as if whenever we downregulate mitophagy, cells induce mitochondrial biogenesis to make fresh mitochondria, and at the same time also induce mitophagy to take down the damaged mitochondria. So they compensate through this SKIN1 mediated mechanism. And if SKIN1 is not there to do this mediation, then of course we have strong effects on lifespan by downregulating mitophagy. Now we can shorten lifespan in the absence of SKIN1 by downregulating mitophagy. And I will finish by giving you some information about what happens actually in neurons when they face oxidative stress. And these are results that we obtained in C. elegans, but we've also confirmed in mammalian systems, in mice and rats. So oxidative stress, when we impose oxidative stress in neurons, for example, in C. elegans, we can do that very easily by introducing specific mutations that affect respiratory chain components, or paraquat. That's another way. It's a herbicide that can be used to induce oxidative stress. In all those cases, what we see is that neurons very quickly cleanse themselves of the damaged mitochondria. And how do they do that? They actually transport most of their mitochondrial complement to the soma, to the neuronal soma, where the degradative machinery resides, the lysosomal system, and most of the autophagic machinery resides. So this is something that neurons do. They cleanse themselves by returning their damaged mitochondria to the soma for degradation. Now this retrograde transport of mitochondria, this trafficking of mitochondria back to the soma, requires calcium is actually triggered by calcium leaking out of mitochondria. This is the most proximal signal. And we can blind these neurons. We can make them not being able to respond to this increase of calcium by using, for example, EGTA, which is a known calcium chelator. And if we use that, then we block this transport, and mitochondria start accumulating in axons, in dendrites, in synapses, because they cannot now be transported. This pathway of transporting mitochondria requires the AMPK kinase and the calcium calmodulin independent protein kinase, kinase 2. This is the kinase that becomes activated by calcium. It's a calcium activated kinase that then phosphorylates and activates AMPK, which eventually results in the retrograde transport of mitochondria to the soma. And there we have stimulation of mitophagy that eventually degrades those mitochondria that are damaged. Now, if mitophagy is inhibited, then mitochondria cannot be degraded in neurons and start accumulating in various appendages, as it happens when we blind these neurons using EGTA, the calcium chelator. And mitophagy downregulation in neurons has devastating consequences for the survival of these neurons and also their resilience against neurodegenerative insults. For example, in this case, we use a model of Parkinson's disease in C. elegans by overexpressing alpha-synuclein, human alpha-synuclein, specifically in dopaminergic neurons of C. elegans. And we can, by overexpressing this aggregation-prone protein, actually cause somewhat neurodegeneration of these cells. But neurodegeneration becomes far more pronounced if, on top of that, we downregulate mitophagy. So without mitophagy, neurons cannot defend themselves. And similarly, what we observed in another model of neurodegenerative disease, Alzheimer's disease, is that accumulation of tau and beta amyloid, two aggregation-prone proteins that have been implicated in Alzheimer's disease. We know that beta amyloid plaques are observed in Alzheimer's disease neurons and also tau neurofibrillary tangles. If we overexpress these proteins, we strongly downregulate mitophagy in these neurons. So these neurons become then defenseless against other neurodegenerative insults. And even in the context of physiological function, mitophagy is required for functions like learning and memory that we can quantify in C. elegans. If animals cannot perform mitophagy, then they don't show this behavioral plasticity that is typical of wild-type animals, which can learn, for example, through associative learning to go towards or avoid certain chemicals if we pair them with food. It's a paradigm of learning and memory similar to Pavlov's dogs that some of you may know. And also in the context of pathology, in the context of Alzheimer's disease, if we upregulate mitophagy, if we artificially now upregulate mitophagy in the context of Alzheimer's disease, where we overexpress tau and beta amyloid, and as I showed you, these two proteins can result in the inhibition of mitophagy. If we now go and upregulate it using specific mitophagy inducers, such as, for example, urolithin A or nicotinamide, these are two inducers of mitophagy, we can restore learning deficits in these animals that overexpress tau and beta amyloid in the Alzheimer's disease model. So I will conclude my presentation by giving you an overview of the pathway that we discovered in worms, which we now also know that it regulates mitochondrial homeostasis also in mammals. So this is essentially a very simplistic representation of the pathway. What happens when we have damaged and superfluous mitochondria? Two things. On one hand, we have oxidative stress that eventually, through a signaling pathway that I didn't have time to describe to you, results in the activation of SKIN1, the transcription factor that controls mitochondrial biogenesis. So we have induction of mitochondrial biogenesis. But on the other hand, SKIN1 also upregulates the expression of DCT1, which is a key mediator of mitophagy. It's a homologue of NICs in humans and in mammals. But this is not enough. So upregulation of DCT1 expression is not enough to induce mitophagy. We need to do this locally in order to selectively eliminate damaged mitochondria and not the fresh ones that have been just produced through mitochondrial biogenesis. How is this happening? It's happening through the activation of the pink parking pathway that modifies, eventually ubiquitinates DCT1, and this is the flag that signals the initiation of mitophagy specifically for the mitochondria that have been flagged, which are the damaged mitochondria. So DCT1 transcription activation is required, but not sufficient to induce mitophagy. We also need, on top of that, the activation, the local activation of the pink parking pathway. And then we also have mitophagy upregulation and elimination of the damaged or superfluous mitochondria. So this is a pathway that essentially controls both processes, both opposing processes, mitochondrial biogenesis and mitochondrial degradation with just one pathway. And this pathway also interfaces with insulin, IGF-1 signaling, and through insulin signaling, again, we can have control of both mitochondrial biogenesis and mitophagy. And for neurons, mitophagy, of course, as I described to you, plays a very important pro-survival role. Whenever mitochondrial damage is detected in neurons, calcium leaks out of these neurons. This results in the transport of mitochondria to the soma, where mitophagy takes care of the damaged mitochondria and inhibits neuronal death. If mitophagy cannot perform this pro-survival function, then we have accumulation of damaged mitochondria. Calcium becomes increased beyond a certain threshold that then initiates a cascade of necrotic and apoptotic pathways that result in neurodegeneration. And with this, I would leave you by thanking my colleagues and friends at the Institute of Molecular Biology and Biotechnology and the University of Crete here in Heraklion in Crete, in Greece. Most of the work that I presented to you today was actually done by two people, Kostas Palikaras and Irini Lionaki. And without their help and also the help of all the other wonderful people here in the lab, this wouldn't have been possible. Thank you very much for your attention, and I would be happy to take questions.